Good. Now it's my time to entertain you again. And I think what I'll do is follow on from several of the discussions today, particularly Dr. Martin's, about what this nutrition might mean in later life. We've been talking about feeding babies more, feeding babies better, feeding babies more aggressively. Maybe sometimes we feed them too much. Sometimes we obviously feed them too little. What does this mean, not just for the acute neonatal period, not just for childhood, but perhaps on into later life? Towards the end of my talk, I will redirect the conversation back to you and me. What if we had been a preterm baby, a growth-restricted baby, an infant of a diabetic mother or an obese mother who also is fat? Maybe I'll even conclude with something that you might use in your own institutions, a political argument for improved attention to problems that begin very early in life. So don't forget my email address for those of you who want to send me a note. I'd be delighted to hear from you. So let's reflect on what is normal growth. We've been talking about this a lot. Growth rate and associated development or functions that occur most of the time. That's kind of obvious. This occurs in fetuses and infants who are not sick or chromosomally aberrant, who consume or are fed normal amounts of normal diets, whose mothers are of normal size and good health and eat a normal diet. If you can find this combination in your neonatal intensive care unit, good luck. Because this is not what we're taking care of. We keep referring to this, but do we really know what this is? Have we ever seen it outside of the womb of a normal, healthy mother during a normal pregnancy? But we can actually have normally grown infants who grow slower than the 3rd percentile or faster than the 95th percentile and end up at any gestational age smaller than the 5th percentile or larger than the 95th percentile, respectively. So small can be normal. Anecdotally, I'll tell you about my wife's family. Her great-grandmother maxed out at 5 feet tall. Her grandmother was 5 feet tall. Her mother is 5 feet 1, protein entering in later on. My wife is 5'2". My daughter is 5'2" and a half and loves to point that out to her mother. My daughter's 2 little girls are both less than the 3rd percentile in height. They both have 50th percentile or above head circumferences, depending on what they're wearing at the top of their head at the time. Now, why should this be important? When my daughter was born, my wife, she was much less than the 3rd percentile. Her head was just fine, right in the middle. Very nice little thing. My wife had another friend who was 5 feet tall, who had a similar little girl born at term gestation, perfectly healthy, being breastfed just as my daughter was being breastfed, perfectly well, doing just fine, happy, just a nice little baby. But my pediatrician saw my daughter with my wife and myself in the clinic and said, "Lovely little child, lovely little girl. I'll put on the chart, small, petite stature, genetic." And everything was fine. But my wife's friend saw a pediatrician who saw this little child who was less than the 3rd percentile, and even though she was a healthy little girl and eating just fine at breast, put on the chart, "If this baby does not gain weight, I'm referring this mother to the Child Protection Agency for not feeding her baby enough." So that's why I talk very seriously. I mean, we have a lot of fun with this, but I do talk very seriously about some babies can be small because that is in their stature by history, by family structure and history. And that is perfectly okay. And I hope all of you talk to your pediatric colleagues who are seeing children after they're out of the newborn nursery and they are small, healthy, normal little babies, and we don't castigate those parents, those mothers particularly, for providing normal breast milk. So to follow on with your comments about let's support breastfeeding, let's do that. And similarly, some mothers are very large and have a lot of uterine size and can produce a very big uterus, a big placenta, and that's going to produce a bigger baby. Now, obviously, there are limits to how big any mother could produce a placenta and a baby, but a big mother, especially if her family structure is such, will produce bigger babies, and there's nothing wrong about those. I could tell you another anecdotal story. My daughter's sister-in-law had a 95th percentile baby, but she is 95th percentile herself, a great big woman, and her husband plays basketball. He's this big. Her baby was eating at the breast and doing just fine. 95th percentile weight, 95th percentile head, 95th percentile length. That pediatrician saw this baby who had not gained weight for a couple of weeks, and also suggested that that mother start supplementing her milk to produce an even bigger baby. So this gets very interesting, and for mothers who are doing their best to feed babies, sometimes kind of sad. But you can also have abnormally grown infants who end up on any gestational age within the normal 5th to 95th percentiles. So if you are supposed to be growing at the 75th percentile in head circumference, length, and weight, and you are losing ground, you're gaining less weight than the normal growth curves, but you are now only at the 50th percentile, that is, in fact, growth restriction. You have not grown at the normal rate. So you can understand how some of these parameters, while we can laugh about some become very serious problems. We really have to keep in mind what we're talking about in terms of growth rates. So, remarkably, in human development, we have two kinds of babies at the extremes. The one on the left is this baby was born in my nursery quite a few years ago. It was a term infant. Mother was an elderly primip, but she was diabetic with chronic vascular disease, hypertensive vascular disease. So she had vascular disease in her uterus, and therefore the placenta could not grow well, and this baby at term weighed 1,300 grams, 1,300 to 1,500, I forget exactly what it is, 1.5 I show there. About the same time, the baby on the right was born, and actually I had to shrink down the size of that baby by picture to get it on the slide. This was a baby that weighed about 6.5 kilos, the largest we've ever seen in our nursery. And this was an infant of a gestational diabetic. Mother had not been known to be diabetic before. She had very unstable blood sugars during her pregnancy, but she never had very low sugars. They were always just up and down associated with meals. And so that high meal-associated hyperglycemia transferred to the fetus and produced meal-associated hyperglycemia in the fetus, and that stimulated insulin secretion. It's that up and down that produces the excessive insulin secretion. And so this baby ends up very large and very fat. Interestingly, later in life, the baby on the left and the baby on the right have the same phenotype. We've been talking about it for 2 days now. The metabolic syndrome of insulin resistance with obesity, type 2 diabetes with glucose intolerance, and various other disorders, cardiovascular disease in particular. So how does this come about? We talk about this in terms of the capacity of human development, and really all mammalian development, to have plasticity, which means while there are normal growth rates, there's great variety that can be produced. The human growth rate can be quite plastic, both in good form and in bad form. So developmental plasticity refers to the ability of a single genotype to produce more than one alternative form or structure or physiological state or behavioral response to environmental conditions. There are forms of adaptation, for example, macrosomia, an adaptation to excessive nutrient supply and anabolic hormone supply, insulin, or there can be constraint, such as the growth-restricted fetus. And most everybody is growing somewhere within these extremes. The mechanisms for this developmental plasticity we speak of as programming, because we're talking about something that happens early in life that persists, producing a phenotype that lasts in some fashion for the rest of the person's life. Programming has been defined many ways. This is Alan Lucas's definition: a stimulus or insult when applied at a critical or sensitive stage. And I've highlighted that, underlined it, because it's that critical, sensitive stage. And I've already brought that subject up before, and I'll come back to it. Of sufficient duration or magnitude. Obviously, a small change may not do much, and a big change for a very short period might not do much. But a significant change for a long enough duration can produce a result that is lasting, perhaps even lifelong, on the effect on the structure, function, or both of the organism. So where did this come from? This is the original data from David Barker that shows from women on the left and men on the right an increase rate of cardiovascular disease and death, and although he didn't include it in this particular figure, metabolic syndrome, as babies were born at smaller and smaller birth weights. Now, if you look clear on the right of each of those, you see a blip upwards. And so most of us recognize the same pattern I've already shown you, that there is a basic U-shaped curve, that very small babies have the propensity for this later life phenotype, just as very large babies do. But this was the original data from a large cohort of subjects that Dr. Barker was able to follow up from early, in England from the early 1900s and come up with this data. So programming, well, he was talking about nutritional programming and environmental programming through the mother's nutrition of the fetus. But this is not a new concept. Programming has been around for a long time. You can use some of these at the next cocktail party or to wow your friends when you go home. What did you really learn at the IPOKRaTES conference? You really learned that if you're a sea turtle, a female sea turtle, and you lay your eggs in a clutch, and those eggs that are in the center are warmer than those in the outside, the center warmer eggs are going to grow up to be little ladies. And those eggs in the outer part of the circle that are cooler are going to grow up to be young men. We have known for a long time that despite our particular genotype, to start with, testosterone is required to produce the male internal and external genitalia. We're all born phenotypically female, and only those of us who have that silly little Y chromosome can produce testosterone and produce this change. So that is a developmental programming. The genes can be different, but only the testosterone, an environmental stimulus, can change the phenotype. We can give testosterone to a female at the right gestational age, and this can produce some abnormal behavior. Makes her more like a man. Well, that's not necessarily good, is it, ladies? You've all read the stories about diethylstilbestrol. The original story showed that this predisposed female fetuses to later life problems, now showing up as genital tract cancers, decreased fertility, increased ectopic pregnancies, and early menopause. I hope those of you who might have been exposed are seeing a physician regularly. This is a serious problem. We've known about teratogenic drugs for years. They alter permanently organ structure and function. Thalidomide is a very nice drug for certain purposes, but it caused foreshortened limbs. Radiation of the thymus was used a long time ago to reduce the size of the thymus in preterm babies with respiratory distress. Shrinking the thymus with radiation was assumed to allow the lungs to expand more. Well, we know now that respiratory distress syndrome has nothing to do with the size of the thymus. Unfortunately, this radiation treatment was, while it didn't appear to have any adverse effects at the time it was given, is showing up as leukemias and early death in children now who are in their 30s and 40s. If you happen to have lack of visual input, cover up an eye, you can produce permanent blindness. So that's called squint amblyopia. And probably the best example that I know of programming is antigen-induced immunity. We're not born immune to external antigens. We are programmed by what environmental stimuli antigens come along. So lots of examples of programming, and I'll come back to some more later on, just to keep that in mind. We're talking more about nutrition, so these are nutritional programming adaptations that persist. I mentioned already Professor Smart's studies in rats that undernutrition at critical developmental stages in fetal life reduces the size of the brain, it reduces brain cell number, and limits the dendritic arborizations, length of neurons, so all kinds of adverse effects that show up later in life with abnormal behavior, learning, and memory. And IUGR fetuses, and I'll show you some of this data, I promised I would come back to that, have reduced numbers of pancreatic beta cells, and so they can't produce as much insulin later in life. And overfeeding infants of any stage, studied in animals and observed in humans, leads to adult obesity. And if that happens to be a female, she becomes diabetic more likely, and as well as obese in her first pregnancy, and even her subsequent pregnancies, and so she can pass this phenotypic programming on to her female offspring for many generations. The longest number of generations I've seen in an animal study was to 7 generations, so a phenotype passed on 7 generations. They didn't go further because they ran out of money. So what are the mechanisms involved with programming? Nutrients and hormones might function as critical signals, acting directly or through coupling mechanisms on sensitive tissues, changing structure or function. So I mentioned already dendritic arborization or glial cell growth. And as you've already heard from the previous talk and others before that, recent MRI studies very clearly show in adolescents who were born small or preterm at birth have reduced brain volumes and the small parts of the brain to go with it. And the few that have been biopsied for one reason or another have the same things that those undernourished fetal rats had, fewer dendrites and smaller, shorter neurons. Then there's a new concept which Dr. New already has presented, and that's called epigenetic programming. I show you a picture here of the DNA structure. That's not altered on the left with epigenetics, but if you happen to change by acetylation or deacetylation the structure, or by other forms of histone modification with various ways that you can do this, you can change the structure of that DNA so that receptor binding doesn't occur. So you can imagine the standard picture of a gene and its active site over here and all of the ways in which that gene can be activated. Anytime you change those activation sites, you can alter the function of that gene. Nothing structurally is changed in the gene, but what it produces is either limited or changed in its capacity. So that's an epigenetic process. And if that happens to occur in the germ cell line cells, now you have a programmed mechanism that will last forever. What about proliferation or adaptive clonal selection? Well, if you deprive a fetus at the right gestational age of protein, and this I assume will happen in a preterm baby of that same gestational age, just happens to be outside of the uterus, so another reason to be concerned about our protein supplies, you can limit the development of the renal glomerular growth process, and so you'll end up with abnormal development of the intrarenal renin-angiotensin system, and this will lead to permanent capacity for hypertension. So those individuals so predisposed, if they eat a high salt diet, if they happen to be highly stressed, their hypertension will be worse as a result. Why is this different? We don't see this when you transplant somebody with a kidney later in life. That kidney can grow and develop and adapt. These kidneys cannot grow, develop, and adapt. These kidneys are simply limited for life. I already showed you this particular observation that if you fast a nursing rat pup, you permanently limit the growth, not just the growth rate, but the ultimate size. You stunt the animal for the rest of its life. Whereas later in life, we can all diet and all gain back the weight that we've lost. I'm going to talk about some examples of each of these two major processes. Constraints, where we have insufficient nutrient supply, and so we end up with undernutrition at critical developmental stages. and talk about the pancreas, and later on in the talk I'll get back to the adaptations in larger infants. This is just a review, something I've already shown you with a little bit more data. You can see in the upper left, when there's nutrient restriction, the fetus will develop insulin receptor activation. So the fetus is trying to upregulate the capacity for insulin to move nutrients from the plasma into cells. That's a natural response. It occurs in all organisms that have been studied, from small little creatures like the C. elegans, the little worms, up to humans and other animals. In the middle, you see glycogen synthase kinase activity. Now, this is a negative regulator. So if you downregulate in response to nutrient deficiency, a negative regulator, two negatives make a positive. So downregulating this negative regulator increases the capacity for glycogen synthesis. And in fact, if we survey all of our IUGR animal models, or you do biopsies of skeletal muscle in human IUGR infants, you see the same thing. They actually have more intracellular glycogen. Whether that's useful or not, we don't know, but obviously this appears to be a developmental response to nutrient insufficiency. On the right, you see P85 PI3 kinase. This again is a negative regulator. So a downregulation of a negative regulator, in this case, will upregulate the capacity for GLUT4, that's the insulin-sensitive glucose transporter, to translocate to the cell membrane. So this is a mechanism that allows you to get more glucose transport for any change in insulin. And then you can see GLUT4 in muscle at least is maintained despite very low glucose and insulin concentrations. GLUT1 in muscle is, but look at GLUT1 in the fetal brain. That's the IUGR fetal brain. It's smaller, but it has upregulated the capacity to get glucose into the neuronal cells, which is fundamentally essential for maintaining survival of those neurons, even though they might not be as prolific or have as long dendrites or as many interconnections. So this allows the normal fetus, the IUGR fetus, in response to very low glucose supply, low amino acid supply, low insulins, to actually have normal glucose utilization rates per kilogram of that small body, despite low glucose and insulins. Now here's the data on the pancreas development. At the top, these are human fetuses. They were IUGR, defined by fetal ultrasound. They had a glucose bolus given to them while they were still a fetus, and then their umbilical veins sampled at the time of cesarean section delivery. These were done by Professor Nicolini and his group in Italy. I don't think we could do these studies anymore. That would be a fairly risky proposition, but he was able to get these done and showed the following results. In the upper right, you see that the normal dark blue bar infants had glucose stimulation exactly of the same degree as the IUGR infants. But in response, the normal infants produced a lot of insulin, whereas the IUGR infants produced very little insulin. Now, if you look at the lower left, you'll see what we've produced in our intrauterine growth restriction model from placental insufficiency, a placenta that's smaller and doesn't transport nutrients to the fetus as well as normal placentas do. And this is a sheep model. And you can see in the dark blue bar lines and dots and lines a fairly good insulin response and a very high insulin level to a glucose load. This is a sustained hyperglycemic clamp. But in the red dots and line, you see the response in the IUGR fetus. So a very slow response of insulin to glucose, and then much lower concentrations of insulin produced. And why does this come about? You'll see in the middle lower panel that those pancreases in the IUGR fetuses simply had very low islet insulin content. And why is there low islet insulin content? That's because there are fewer beta cells within those islets. They are smaller islets, and instead of having lots of beta cells, there are few beta cells. And that's because there's a programmed mechanism of decreasing cell replication. So at some point in response to low energy supply and low protein supply, the beta cells shift from producing more beta cells to fewer beta cells. Why would they do this? Well, teleologically it makes sense, because if you have less nutrients, why would you have an anabolic hormone? You don't want to use those substrates to grow. You simply want to allow those nutrients to go into cells for normal metabolism, just to try to maintain survivability. There's the story again about the growth factors. So you really don't grow as well when you have persistent reduction in nutrient supply, particularly amino acids. In the upper left, you don't have a growth factor that allows for mitogenesis. So this probably is what we'll see in our pancreas cells, one of the factors that may lead to failure of those pancreatic cells to divide and replicate. mTOR and EIF4E are the factors in the insulin signal transduction pathway that activate the synthesis of amino acids into protein, and those are both down with chronic growth restriction so that you can't grow as big a muscle. And the binding protein for EIF4E is obviously increased in this case, so that's a double jeopardy. That protein is simply not as available to allow insulin to stimulate protein synthesis. So this decreases the capacity for growth, allowing whatever nutrients can get across the placenta to be used for metabolism, where there is an upregulation of capacity for energy uptake. In addition, we found some evidence that there might be an increase in some protein breakdown. So I've shown you insufficient glucose and amino acids leading to slower rate of growth. And this is data showing that there are muscle-specific ubiquitin ligases that change during nutrient restriction. You may not come across these proteins every day in your neonatal intensive care units, but the one on the, whichever one you're looking at, that's the control value. With hypoglycemia, that ligase goes up, and it's recovered when you get more nutrients back, especially glucose, and the same for this other one. So what you're basically doing here is showing that chronic fetal glucose deprivation increases these ligases, which in turn allows for greater amino acid release from fetal tissue. So you not only are not growing, but you are also increasing the capacity to break down protein to provide amino acids for probably glucose production, or at least amino acids for oxidation, so that you maintain energy status. So we end up with this IUGR fetal phenotype, which I've explained already. Just to review, we have increased capacity for glucose uptake and perhaps increased insulin sensitivity. I've shown you now the data for decreased pancreatic growth, development, and the capacity to secrete insulin. And we also have the decreased capacity for amino acid synthesis into protein and for cell growth, and increased cell protein breakdown. So what if these patterns persist? What if they don't just stay around at in the very early fetal period, that they actually are programmed by these epigenetic processes to last for later in life? Well, if they recur, if they last, and you happen to be an infant who then eats a high sugar diet, what's going to happen to that sugar? It's going to be taken up very rapidly into cells where it's going to produce, along with the glycerol and the triglycerides that are around, because insulin isn't doing much with them, it's going to produce fat cells, more fat cells with more fat in the cells. And so you end up with obesity, insulin resistance from the obesity. Started with insulin activation, now we have insulin resistance, reduced capacity to produce insulin, so you have inhibited insulin action, but you can't compensate by producing more insulin. You have fewer pancreatic beta cells. You have reduced muscle mass, so you have nowhere to put the glucose, even if you had some insulin and some insulin activity. So you become glucose intolerant, and that's called diabetes. And you're also short, as several people have already shown. So what about the preterm baby on the left? How are we going to feed it to get it to look like the baby on the right and not the one in the middle? We've already talked about these problems that we are not feeding babies sufficiently and they're not growing fast enough. We've talked about the insufficient protein leading to growth failure. that if you're born at the comparable period from these fetal sheep to fetal humans of around 24 to 28 weeks gestation, you end up, we need a lot more protein than we're actually feeding these. Dr. Ziegler's method showed 4 grams per kilo per day. This animal data, when transferred to human growth rates, says the exact same thing. We need about 3.5 to 4.5 grams per kilo per day. Here's the short stature. The dark bars, these are 17-year-old girls. Same picture for boys. This is Maureen Hack's data that shows the shift to the left, meaning shorter stature. More of these girls that were preterm and growth restricted are now shorter when they are adults. And I've shown you already this interesting data from Sudha Kashyap and Bill Hurd a few years ago, where if you feed more protein, you will get a larger head circumference, a longer length, but no increase in fat mass. And if you add to that higher protein intake, or any protein intake, excess energy, you produce an increased amount of weight. But that's not due to an increased head circumference and brain size or increase in length. It's due to an increase in fat mass. So we definitely can influence the growth rates of these infants. Several of us keep coming back to this concept that, and Dr. Martin went through this in great detail with you, about how selected fatty acids, those that are fundamental for forming the membrane sheaths of neurons and allow those neurons to grow and function normally, if not provided at critical developmental stages, don't end up with normal amounts of these neurons and normal functioning neurons, so that we actually can see neurodevelopmental abnormalities in infants that do not get sufficient amounts of particularly the docosahexaenoic acid, the omega-3. Less visual acuity, improved Bailey-Mental developmental and, higher visual acuity and improved developmental scores when fed to more normal rates of supply of these particular fatty acids. And again, just to remind you, the good news is that better nutrition produces not just larger brains, but functionally larger brains, so that intelligence quotient improves directly with the growth of the brain if we actually go ahead and feed these babies more. Several of you have asked in response to my statement the other day that, and we had another discussion already this morning, about the growth-restricted infant, what do you do to re-nourish that baby? And this is an area of research. I wish we had the answers here. In the upper left, just to remind you that when you acutely deprive a fetus or a preterm baby, or you or me, of nutrients, particularly energy, we break down our protein, we oxidize those amino acids, that maintains our energy supply. That's not a good long-term strategy to just disappear in order to keep a few cells going. So over time, that's the next two bars, chronic hypoglycemia and hypoinsulinemia, the growth rate slows to conserve amino acids for what little metabolism and growth they're needed for. And that allows energy metabolism in the cells to normalize. And so these infants are adapting chronically to this low energy and protein supply, and their capacity for growth might be limited. Feeding these babies even the normal expected amounts of protein and energy at their gestational age, whatever it is, might not be as successful. And I leave this as something for research. And we're actively working on this in our intensive care unit, in clinical studies, and in our laboratory right now. This particular study I found very interesting, just published in Pediatric Research. Intrauterine growth restriction in the absence of postnatal catch-up growth. So these infants were not fed aggressively. They were IUGR, but they were not fed high protein, high energy diets to try to catch them up, as if despite chronic adaptation with slower rates of growth to producing the growth restriction, they were fed to see if they could get them back to the normal growth curves. In this particular case, there was improved whole body insulin sensitivity in the offspring if they were fed a low protein diet. So maybe if you're growth restricted, the best way to take care of you is to leave you slightly growth restricted. The alternative is to get that adult, obese, insulin-resistant phenotype much earlier in life. And the studies, I think we've already mentioned several, that are showing more aggressive nutrition of growth-restricted infants that are chronically adapted to growth restriction are the ones that get fattest soonest. Could we do anything about this in the fetus? So none of you is an obstetrician, is that right? Are there any obstetricians out there? Well, this is where I keep saying, why is a neonatologist doing this research? Where are my obstetrical colleagues? Why aren't they trying to correct growth restriction when they see it? They first identify growth restriction when the fetus is still alive inside the womb. Well, actually, there have been some trials, not by obstetricians, by pediatricians. These are reviewed in this nice article. I'll give you the reference another time. So, 2 trials, 1,076 women, high protein supplementation. What did this do? These women were identified as being at risk for growth-restricted fetuses, either because they'd already had one, or in a previous pregnancy, or because they were undernourished. They were in a clinic where undernourished women were being seen during early pregnancy, and they were recognized as not having good fetal growth already. And they said, let's try to improve their growth and give them the growth of their fetuses and give them a high-protein diet. And what happened? In 3 of there was a significant increase in the risk of small for gestational age, meaning IUGR infants. So the exact opposite of what you wanted. They gave high-protein diets to the mother to try to correct this growth restriction in the fetus, and they got worse growth restriction. Then there were 3 trials involving almost 1,000 women who were given an isocaloric protein supplementation, and this was associated with increased risk of intrauterine growth restriction, producing small for gestational age infants. So almost 2,000 pregnancies. I think that's pretty convincing. Basically, this kind of data stopped research in this subject. They said, look, giving more nutrients, particularly more protein, to women who are already carrying an IUGR fetus is more damaging than it's worth, and let's just not do it. So not only did they stop doing these nutrient supplementations, but the obstetrical community stopped doing any research at all. So not allowing my obstetrical colleagues to rule the day, we're going about this research in a fairly rigorous way using an animal model. These are data from our experimental sheep. What we did was infuse trophamine into the pregnant ewes, into the pregnant sheep. And you can see that the essential amino acids are more easily transported across the placenta. So this is umbilical or fetal glucose, fetal uptake of these amino acids, and this is the change, the increase in the amino acid concentrations in the mother. So we had trophamine being very high, enriched in essential amino acids, showed that the essential amino acids, particularly the branch chain amino acids, leucine, isoleucine, valine, more easily crossed the placenta, and there was a larger delivery of these amino acids into the fetus. So there is, even with the growth-restricted placenta, there is increased potential to transfer some of the essential amino acids into the fetus. So that gave us some encouragement that there might be a way in which we could get more nutrients to a fetus, and if we could identify a growth-restricted fetus before that nutrient restriction programmed it irreversibly, we might have a shot at improving things. My obstetrical colleagues were...
That's what I asked Kami before.
Yes.
If we give to the mother more nutrients,
can we do anything?
well, the baby will be better.
Well, if you catch the baby, we think, before growth restriction is programmed, we might have a chance. So unfortunately, my obstetrical colleagues laughed at us because we got the same results. But we actually have data now that we can work with. In the upper left, what we saw was an increase in response to this nutrient supply to the mother. We gave the mother more trophamine, got more amino acids going into the fetus, an increase in metabolic acidosis in the fetus. We saw hypoxia develop in the fetus, and we've seen the same data by giving glucose. There was hypercarbia. The pH was lower, the O2 content was lower. In response to the hypoxia, there was an increase in the production of red blood cells. So could we go about this differently? That's high protein. Let's try high energy. And in fact, if you give an energy supplement along with protein, so this is not the protein only or the isocaloric protein supplement, this is a high energy supplement. 13 trials involving 4,500 women, balanced energy protein supplementation produced a substantial reduction in the risk of small for gestational age infants. And you say, great, that's what we should just do. Feed the mothers more sugar. Feed the mothers more fat. Put them on a Twinkie diet. Do you know what Twinkies are? Oh, come to America. We will show you the worst of these. It's a little glob of something that's very creamy and very sweet, and it tastes wonderful, and it's pure sugar and fat. That's a Twinkie diet. Well, what they didn't do was measure head circumference and length. So I assume what happened here is that these babies were not SGA, not because their heads were bigger or they're longer, but because they were fatter. And that makes perfect sense. And everybody that's studied this now has found the same results. A higher energy diet to the mother, whether she has an SGA baby or an IUGR fetus or a normal fetus, produces a fatter baby. So this is simply fat production, and we know that's not good for you. So does this mean that energy is good? Of course not. So we tried to do this anyway, just to follow up on our studies by giving the mother the pregnant sheep trophamine. We gave the pregnant sheep a dextrose infusion, and she became hyperglycemic. And sure enough, when we looked at the fetus, we saw the same problems. The low PO2 went down, the PCO2 went up, the pH went down, the saturations and blood oxygen content went down. There also was an increase in lactate. Not surprisingly, more glucose is metabolized to lactate. And with that comes the obligatory protons, so they had a metabolic acidosis and their pH went down. So we're still losing. But we have one more set of studies that we're going to be doing this next year. Hypoxia, that happens in both of these cases, high energy or high protein. So what are we going to do? Well, we're building an environmental chamber that will allow us to produce hyperoxia, so the mother's breathing more oxygen. Don't think we'll get very far with that because mother has normal oxygen to begin with, and the fetus is not hypoxic. Its PO2 is lower than the mother's, but its blood oxygen content, at least in the normal condition, is just fine. So we don't think we'll see much of an effect of a higher PO2. But that will allow us then to compare the high oxygen environment from higher PO2 with a separate set of studies in which we will transfuse the fetus. We'll have donor fetuses and we'll have study fetuses, and we'll take blood from the donor fetuses and produce a red cell transfusion into our study fetuses. So we will increase red blood cell mass, blood oxygen content as a result. And remember that study I showed you the other day of the fetuses that were in mothers, pregnant sheep that went to high altitude. Those that grew normally had higher red blood cell mass and higher blood oxygen contents, or at least maintained normal blood oxygen contents. So hypoxia by itself increases catecholamines. This reduces insulin secretion and insulin action. It increases cortisol, which will increase gluconeogenesis and will increase protein breakdown. So we're hoping that by blood transfusions, red cell transfusions into our IUGR fetuses, we'll get past this problem where the protein supplementation or the energy supplementation or both of them together led to hypoxia and these adverse effects, not just of failure to grow, but of increased perhaps protein breakdown. There's good potential that we might also, while we're doing this, increase insulin production in the fetus. So hopefully we not only will produce better oxygen content and reduce the catecholamines and cortisols, we'll also increase insulin production. So we'll have the best, or at least the best possible world out there. Here's 4 amino acids, isoleucine, valine at the top, arginine and cysteine at the bottom. And you can see a very steep relationship between insulin concentration and the concentration of those amino acids achieved by our dose of amino acids into the mother. So even though the fetus got hypoxic, there was still an increase in insulin production. So there's some potential out there, and I already showed you that we may be able to get some myoblast development here where we actually increase the replication of cells, since this occurs in vitro. This is in vitro where we don't have catecholamines, we don't have cortisols, and they're in normal oxygen environments. So there may be some light at the end of the tunnel, and if you invite me back in a couple of years, I might be able to tell you a much nicer story about how your obstetrical colleagues could put you out of business. What about adaptations? This is overfeeding. What do we do in the newborn nursery that produces excess fat? So the opposite of what we've been talking about almost solidly for 2 days, where we've been talking about trying to be more aggressive to prevent undernutrition. If we give too much carbohydrate and fat to any mammal, whether it's a human or a rat or a mouse or a sheep, we produce more fat, and that just keeps on propagating so that we end up with more fat later in life. If we produce a high glucose load, we end up, as I've shown you, with insulin resistance and glucose intolerance. That's excessive glucose infusion, so our TPN supply should not be producing glucoses above 120 milligrams per deciliter, or you'll end up perhaps doing you more harm than you thought you were helping with. Inhibiting or enhancing insulin secretion from different degrees, patterns, and duration of hyperglycemia caused by excess intravenous glucose infusion. So if we keep that glucose concentration very high, you can suppress insulin production. If you have this kind of up and down insulin or glucose supply, you may produce more insulin. So we have to pay attention to how we're managing our glucose infusion rates. Simply asking the nurse to start the IV at a certain rate does not guarantee that that pump delivers a constant supply just as we want it. And so I do encourage you to start looking into continuous glucose monitoring as a method to find out in your own nurseries how you are doing. Every time we've looked at this, and those who are using the glucose monitor, this continuous glucose monitors have looked at this, they're finding much more variable glucose concentrations than any of you might have imagined. And one of our colleagues here today has been doing these studies and can talk to you about that personally. Overfeeding in childhood, I'll come to that in a minute. And overfeeding producing more rapid weight gain produces more obesity. And this side I've shown you, and others have commented on this, whether you're born small or large, small babies and large babies that grow faster than they should all end up with a higher amount of body fat later in life. So we can induce obesity by more rapid weight gain, whatever size you're born at. And we do that fairly obvious. Now, is this a problem in the newborn nursery? We don't see it that much. Is it a problem in early childhood? Well, the obesity that we see in early childhood that comes from this portends later life problems. As these infants grow up, their risk for coronary disease and stroke magnifies. It increases exponentially. The risk for these two serious disorders increases exponentially. And perhaps these were the two disorders that led to Dr. Barker's observations of increased mortality at earlier adult ages in those infants who were born too small. And he now agrees, of course, that if you were born too large, you can have the same later life complications seen in type 2 diabetics. So what do we do to our children after they leave the nursery, or even if they were normally born, and going home to the normal American environment? We're trying to sell you the normal American environment. It's called Twinkies, perhaps. What do you get at Starbucks? It's not just the coffee, it's everything you put in the coffee. And don't forget Pizza Hut and McDonald's. And we're really providing the rest of the world some of the greatest poisons out there. What do we do at home? This is just a nice survey from not too long ago published in Pediatrics. Look over there on the left. These are children. These are little babies. These children aren't even 1 year old. And 3 to 7% of them are eating candy, chicken nuggets, fried chicken, snacks, chips, popcorn, cheese puffs, pizza, sodas, sweetened teas, hot dogs, sausages, cold cuts. My gosh. And look at 19 to 24 months. Up to almost a third of the infants in the U.S. are eating this horrible diet. candy, chicken nuggets, fried chicken, snacks, chips, popcorn, cheese puffs, pizza, sodas, hot dogs, sausages. My gosh. This is what we need to do. You have a very lovely example here, our breastfeeding Mother of the Year, our poster breastfeeder that's been helping us with the meeting. Breastfeed for longer than a day or two. Breastfeed for infancy, whatever do you define for infancy. The growth curve on the bottom is the breastfed infant, and I hope all of you are now using the WHO curves for normal weight gain during infancy and early childhood from those that are, it's data derived from mothers who exclusively breastfed. And that's a much better growth curve from those chicken nuggets supplemented formula-fed infants that you see at the top there. Now, I said I'd bring this back to you and me. What about later life? Because what we do later in life is try to compensate for the earlier part of our lives, and it's very difficult to do. Those of you who need to lose weight and those of you who just want to stay slim and trim have to do a lot of different things that are very difficult to do. There's no question about it. Dieting is not easy. Food tastes good. Staying alive is fundamental to human beings, to all animals, and so we eat. So not eating is just darn difficult to do. And perhaps some of us were programmed to have less leptin activity, so we don't have the capacity to lose our appetite when we eat. We still have our appetite when we eat. Or we exercise. We get out there on the treadmill and we sweat and we run outside and we just keep on jogging. And we do this with a lot of pain and injury. And that's the lifestyle that we try to lead in order to stay slim and trim or to lose weight if we've gained some. These graphs all show you the same thing, that exercise and caloric restriction are necessary because just a reasonably healthy lifestyle, meaning being free of illnesses and eating a reasonably regular diet, just doesn't cut it. No change in the truncal fat mass or the abdominal fat mass in those who had a just normal, reasonably healthy lifestyle. You had to go out there and struggle on the treadmill, or you had to go out there and just not eat what your friends were. And your insulin sensitivity index, shown on the left, or your glucose area under the curve on the right. Exercise and caloric restriction. Tough stuff to do once you are programmed to be whatever you are. That's really hard. So I can't tell you that this is just going to, healthy lifestyle won't do it. You really have to work at it. The good news is that there's a benefit of caloric restriction versus the extent of restriction. These are studies in rats and mice. So this is extent of lifespan increase. And you can see that for the extent, as the extent of calorie restriction goes up, so this is the extent of calorie restriction, we can extend our lifespan. Now that's data from rats and mice. You can see it in the smallest of organisms, the mice, the flies and the worms. Will it apply to humans? There is some intriguing evidence out there now that a lean diet, a lean, healthy lifestyle, meaning keeping active, and not having been programmed to obesity, insulin resistance, and the complications leading to diabetes, actually can lead to a longer lifespan. And the earlier you start, the better off you are. Good news is that's for those of you who are young. You can do something about it. For my age, there's nothing more I can do. So that's the point of that's the percent of my elapsed lifespan, and I'm out here. It's all over. So I get to go eat sausages for breakfast because it's not going to help me. Whether I gain or lose, it's not going to matter. Just to bring you back to the point that there's lots of other examples of programming out there, and I'll get to the political argument in a moment. So we talked about iron deficiency leading to cognitive delays. Mike Georgiev's studies have been really seminal in this regard in recent years. A nice study showing reduced somatic and head growth contributing to neurodevelopmental delays. This is just one more. Dr. Martin presented several other examples, as Dr. New did. I'll give you an interesting example from my own institution. If you happen to be unlucky enough to have schizophrenia in your family, so you're predisposed to being a schizophrenic, the problem schizophrenics as children have is that the first thing they cannot do is tune out the environment. All environmental noise and light and activity and all sorts of stimuli, they can't dumb that down. They can't tune it out. That's because they do not have the negative feedback in their in certain neuronal pathways, so that a stimulus going through tends to produce a negative feedback, and this more stimulus, the more negative feedback, so you actually start restricting the environmental stimulation. What makes that pathway develop a negative feedback loop? Turns out it's acetylcholine. So one of my psychiatry colleagues is giving pregnant women in a very controlled study from sponsored by the NIH choline supplementation to see if he cannot first prove that it's safe, and then secondly see if he might find, as he has shown very clearly in an animal model, an increase in the capacity for neuronal suppression of stimulation. Really neat study. Many studies now in all sorts of animals showing that if you give the mother who's pregnant alcohol, her offspring, even as immediately after birth, will go to alcohol preferentially. So if you put cups of fluid in front of this little neonatal rat, just born, and one has milk, neonatal rat pup goes to the milk. If you put milk that has alcohol in it, rat neonatal pup goes preferentially to the milk that has the alcohol in it. If you put water, rat goes to the milk. If you put water with alcohol, the neonatal rat pup preferentially goes to the water that has the alcohol, bypassing the milk altogether. I blame all of my alcoholic tendencies on my mother, who obviously must have had some drinks during pregnancy. Really neat study. We all know about intrauterine exposure to drugs, cocaine, alcohol, tobacco, and marijuana. These all have been associated with reductions in head circumference and performance intellectually later in life. Early childhood neglect, not just undernutrition, but specific neglect in the first 2 years of life is a precursor for later life aggression. So behavioral developments can be a programmed response to a behavioral insult earlier on. If you give antibiotics to your babies in the newborn intensive care nursery, no matter what their disorder, even if they never had an infection, those infants later in life have increased reactive airways disease and will wheeze when they exercise. So think about that the next time you do a rule-out sepsis. Baby's not truly sick, but you're concerned about risk for sepsis, and you give 2 to 3 days of antibiotics. And we all know the folic acid story. If you don't give enough folate and the mother's folate stores are low, there's an increased risk of neural tube defects. But if you give too much folate, there's an increased risk of asthma. So there's lots of balances about too much versus too little. So now for the political argument. This is a figure that came from Dr. Elias Zerhouni, who was the former director of the National Institutes of Health. And when he presented the case for the reauthorization and refunding of the National Institutes of Health in 2007 to both the House and the Senate in our Congress, he used this curve. What he suggested was that if we find out the mechanisms, he used the word molecular, but he's basically talking about the mechanisms, whatever they are, and you could solve how a disease got started, you could preempt the development of that disease, and this exponential increase over the lifespan of that disease, of the financial cost to that individual and to society, and the disease burden to the individual and the disease and the individual's immediate family or those who take care of him. A nice concept. Preempt a disease, prevent its complications, prevent the cost of those taking care of those complications, avoid all of those things our internist colleagues do every day, taking care of us, taking care of hypertension, taking care of diabetes, and then reduce the disease burden and make that person a more productive citizen and obviously contribute more economically. We'd all have more better economies if this were the case. Now, the disease that Dr. Zerhouni used as his example was rheumatoid arthritis. Now, that was probably appropriate for our Congress because rheumatoid arthritis usually begins in 40s to 50s, and all of our members of Congress are at least that age, and some are almost twice that age. So that was perhaps an appropriate disease. And I liked the concept, so I just said, wait a second, I'm a pediatrician. Not just a pediatrician, but a neonatologist. And not just a neonatologist, I've been studying fetal development my entire career. Let's just shift this curve over to the left. And so I came up with this concept. It is published. You can read about it in pediatric research. The unique advantages of research in early life. And I hope you'll sell this to your political colleagues. Preempt lifelong diseases that begin early in life. We've talked about preterm birth, genetic diseases we haven't covered at this session, but we have spent a lot of time on IUGR, and I've touched a bit on fetal obesity as well. We brought up the issue of all sorts of inflammatory disorders, the impact of antibiotics on taking care of not just sepsis, but inflammatory disorders, even though we didn't know that there was just inflammation and not sepsis. We've talked about undernutrition and the malnutrition that's associated with that. And I brought up some concepts about drugs, bad drugs. And these are going into the pregnant mother and the fetus and the early newborn infant. If we could preempt any one of those in some fashion, restrict their development, that's the kind of restriction we should really be trying to develop. Over the lifespan of the infant growing to a child, to an adolescent, and to a young and older adult, we might be able to prevent the lifespan of disease burden and the financial costs that go with these. Basically, this is the story of pediatrics and what it's all about. All of us know that if we could do better for our children, we could improve their lifespan and improve the health of our populations as well. And I'd be happy to send you that reference so that you can take that to your local politicians and tell them to fund your research programs, your neonatal intensive care units, and maybe even to educate your obstetricians. Thank you very much. That's a good insight for a good thing to move us on to caloric intake for a coffee break. Questions?
I would like to thank you very much for this exciting lecture. I would like to come to one of the studies you mentioned, the very recent studies, where the IUGR baby was not, did not have catch-up growth, and they had a better development later. I would like to ask you, if they don't have a catch-up growth, if you don't provide them the materials to have a catch-up growth, this might influence his or her brain, you know, what we extensively discussed in this seminar. Would it be better to give the nutrients to this baby so that he or she has a catch-up growth, but simultaneously have exercise, the lifestyle of this baby and the exercise, not the baby, the child, yeah, just to balance the nutrients that you give to this in order to catch up growth, and on the other hand, have from the very early life exercise?
Yeah, it's really an excellent question and gets to the issue of really what should we do with the infant already IUGR with established chronic adaptations. That one study is a rat study, so we don't know if this applies to humans. So first off, let's be cautious about interpreting what might happen to humans from another animal study. But it is intriguing. I think we have to reflect on two possibilities here. One is that you could provide the right kinds of nutrients to promote good brain function and development as best they can be developed in a baby that already has fewer neurons, shorter dendrites, and fewer interconnections. Do the best you can. So we've talked about certain nutrient factors, not just total protein or total energy, but improving docosahexaenoic acid, or continuing research into what is it in breast milk that promotes better neuronal development and behavioral and cognitive outcomes. And substitute that, or at least balance that, with a shorter stature. Does it matter if you're short, if you're smart? So that's just one concept. The other, of course, is to say, let's do everything we can with all kinds of nutrients and promote a better brain as best we can and allow that individual to have some other disorders, not short stature, but the metabolic syndrome to whatever extent it develops in a particular individual. Now, I think those are two options, and I don't know which would be the best option. What would you choose for yourself? What would you choose for your child? And if you're a pediatrician recommending nutrients to a mother, what are you going to tell her to do? Because basically it's this infant, and that's really what I was trying to get at with that lifespan growth curve. We're doing things in this infant which we would like to have help the infant over its lifespan. We're not giving the infant the option to choose whether it wants to be diabetic but smart or short but smart. We hope that those are the only two options. We might, in fact, I think as I showed you from our research studies, actually cause harm and make things worse by one of these more aggressive approaches. So I think the issue about how do you manage the growth-restricted infant is really, for me, the most important. I think we've really presented more than sufficient evidence that if the baby is simply born preterm, then there's every reason to provide the nutrition that baby needed and should have gotten if it were still a fetus, so that we maintain normal growth and development as best we can. But if that baby, either because of fetal problems such as placental insufficiency, is already chronically adapted and growth restricted, or we chronically produce that adaptation by underfeeding babies in the neonatal intensive care unit, then what do we do? And that's the area that I think is still unknown. So I'm glad you brought it up, because once we start seeing these studies that tell us there are options out there, you could maybe improve with selective nutrition neuronal development, but not gain weight. Maybe that's not so bad. Maybe be a little shorter. Maybe that's not so bad, as opposed to developing the metabolic syndrome, which really is a very costly outcome for an individual and for society. So I'm glad you brought that up as a really intriguing proposition. Yeah.
Thank you for your excellent talk. Just to make things easier for us, when we have IUGR babies in the unit, what would you suggest us to do? Calculate IV fluids and calories and everything according to the weight that they are born, or to the 10th percentile, and which curve would you like to see them to follow after birth? Being on the same curve, trying to enter the curves, the regular curves? What would you suggest to us?
Really good question. This is what, every time I give this kind of a talk in shorter fashion to the residents on rounds, that's the first question they ask me. So I think two things. If you're trying to provide glucose by intravenous infusion, take the size of that baby's head and take the 50th percentile weight for that head size and use that weight to calculate your glucose infusion rate. It will be higher than using the body weight. And that's good because it's the brain that you're trying to give glucose to, not the fat and the muscle, which are already small, and in fact, as I've already shown you, have a capacity to take up more glucose than they need. The brain needs that glucose supply, so that's for glucose infusion. Now, what about protein? I would stick with the body weight because it's the mass of the body that determines protein requirements. Your bone length and your muscle mass are what contribute, other than water, to most of your weight. And so I would stick with that for your protein, whether it's enteral or parenteral. What growth curve would I choose? I again would choose the growth curve appropriate for that length and head circumference and weight on balance, so that you're not trying to over-nourish this baby. And I say that if you are really dealing with a growth-restricted infant. If it's just a preterm baby, then you would like to be sure the baby's getting what we talked about as aggressive. But if it's growth-restricted, then I would stick with the requirements appropriate for the size of the baby. Only thing I would change is use the 50th percentile weight for the brain to maintain a higher glucose supply for the brain.
Only glucose.
That's only glucose, yes, not for protein or any of the other substrates.
Thank you.
Yes.
May I have a question. Do you think it is better to give in all babies when the mother's milk is not enough, or when the baby don't take mother milk, extensively hydrolyzed milk, because this memory programming you mentioned in...
I'm sorry, I don't understand.
There are guidelines in high risk for atopy families in the newborns to give breast milk or exclusively or extensive hydrolyzed milk. Do you think it is better to give to all babies extensive hydrolyzed milk, maybe in the first days of life? Thank you.
I see no advantage to hydrolyzed proteins. There's a theoretical advantage for reducing allergy, so you don't have intact proteins. But I don't think there's any advantage nutritionally. In terms of, I think the other part was like hind milk, milk that has more fat in it, from the mother's milk, that will produce a fatter baby, but it doesn't produce more nourishment. I'm not sure I'm answering your question, but I think hydrolyzed protein to a mother who's trying to establish feeding, I don't see any advantage to it. I think this is an area of further research. We've had several other questions about this. To me, it's theoretical that you might reduce allergy, but I don't think the hydrolyzed protein produces better nutrition. So I would start first with Joe New's concept, and others have talked about this, that anything you put in the gastrointestinal tract seems to promote villus hypertrophy and enzymatic development. And it doesn't matter what it is, because your goal is not nourishment, your goal is to stimulate growth and the production of the enteral hormones that are anabolic to the gut.
Thank you for your, thank you for your excellent conference. I would like to ask you, are there any metabolic markers in IUGR babies that can help us to distinguish IUGR babies from SGA babies? I mean, babies coming from small parents. And if we can follow these markers, you know, given the proper nutrition to make them normal, for example.
Yeah, the best markers that I know of are the size of the mother. The paternal influence is on the placenta, but this has been studied many times in many different kinds of animal models, that if you take a big fetus, an embryo from a potentially big fetus from a large animal and put it into a smaller one, you get a smaller outcome. And vice versa. If you take a multitocus species, so take a rat that has 7 fetuses and you take 3 out, the other 4 will grow to their more maximal capacity. So as far as I know, it's the size of the mother that matters, and that's the marker that I use along with intrauterine growth. Now, could we improve measurement of intrauterine growth? Those of you who have been pregnant, and that's none of the men, you will know that your obstetrician took out a tape measure and measured the fundal height, and that's just not a very accurate measure of growth of the fetus. It depends on your body habitus, how obese you happen to be, or how thin you happen to be. It's just not very accurate. So should we recommend serial weekly ultrasounds in all pregnant women, or every other week, or at least monthly, and compare those to established growth curves that we obtain in different centers by serial ultrasonography of normal pregnant women so that we have normal fetal growth curves. Well, that sounds like a nice thing to do, but there's a lot of argument against doing it. Ultrasound has increased in frequency over the years. Perhaps it's time to revisit that. I know the American College of Obstetricians and Gynecologists revisits this almost annually to try to decide whether they should recommend serial ultrasound in everybody, not just at 2 gestational ages to try to guess at gestational length. So those are the best guides I know. I wish there were some biochemical measurement or hematological measurement, but I just don't know of any.
To this question, may I tell you that we use the GROW curves of the GROW program of Cardozi, which takes into consideration the intrinsic growth potential. The mother stature, what you said, booking weight and height, the parity, the gestational age, the gender of the fetus, and ethnicity, the altitude at which the pregnancy evolves. So this one gives you the intrinsic growth potential. And if you have normal Doppler studies during pregnancy, then you know, and then you have the customized centile of the baby.
Yes.
And this customized centile, if it is below 10%, and this is very likely to be IUGR and not SGA.
Yeah, I think that's an excellent point, that there are many approaches. That's one that I've read about, and very, very good one, to try to look at all of the factors that might contribute from the maternal perspective to the growth of the fetus and make a good estimate, a better estimate. When you actually look at if they break out each one of the factors that they put into the Cardozi study, you still find that mother's height, her family history of height, meaning her mother, her grandmother, her great-grandmother, and her adiposity and weight gain during pregnancy. So pre-pregnant adiposity, not just her weight. You could use BMI, but some measure of adiposity, and the gain in adiposity during pregnancy, not just weight gain, because that can be due to water and salt as well. So fat before birth, or excuse me, fat before pregnancy, fat gain during pregnancy, and the mother's height and stature throughout her family history. Those account for 95%, almost 95% of the impact on the size of the fetus at birth. So those, again, are the most the key elements. I still think it's best to come back to we need a better measure of individual fetal growth, and maybe somebody will come up with it. Yes? Oh.
Thank you for your excellent lecture. I would like to share some of my thoughts about the oxygenation status of the pregnant mother, as you mentioned. I suppose this is, you're formulating a hypothesis, not yet established anything, but still, most of us, if I'm not mistaken, consider the IUGR fetus a hypoxic fetus. And in most of the cases, especially when the Dopplers are impaired, for example, in reverse and diastolic flow, mother is normoxemic and the placenta might be normoxemic, and then the fetus, in most of the cases, is hypoxic. So how possible do you think it is for us to depend on this hypothesis, that if we hyperoxygenate the mother, we will be able to hyperoxygenate the fetus? This is the first question. And the other one is about, you mentioned the implications of the oxygenation on cortisol and catecholamines. Because we have done a series of studies on angiogenesis and IUGR, I'm just thinking that the oxygenation status of the fetus might be implicated with also angiogenesis. Because, for example, it is established that VEGF is downregulated in hypoxia. So it might be an interesting hypothesis to see not only how oxygen is implicated in catecholamines and cortisol, but also what it does in certain angiogenic or anti-angiogenic factors of the fetus. Thank you.
You just covered the next 3 grants that I'm writing. That's just wonderful. I don't know how I could respond to all of that. You're absolutely right. This is a hypothesis. It's based on the observation from the fetal studies of the normal fetal lamb raised at altitude, that those that produce more hemoglobin, more red blood cells, had normal growth rates. Could we take a fetus that's already mildly hypoxic, and this comes from placental insufficiency, not because mother's hypoxic, and increase red cell mass, improve blood oxygen content, and prevent the chronic adaptation to hypoxia and achieve better growth, prevent the increase in cortisol and catecholamines that we actually can measure in our IUGR fetal animals? Well, that's, as you say, a hypothesis. What about the angiogenesis? We are actually looking at this. So what leads to that beta cell, the failure of beta cell replication in the pancreatic islets? It looks like the vasculature is reduced first. So if you go back to the earliest stages of our IUGR model, before we see any change in the number of islets, the size of the islets, or the number of beta cells, we see a reduced vascular bed in the early developing pancreas. And that just does not continue to grow. And then you see the reduction in pancreatic islets. Now, that could be sequential and therefore causal, so that angiogenesis might be a critical feature of producing this abnormal beta cell development. Or they might be parallel, the same insult. The lack of oxygen, the lack of amino acids, the lack of glucose, they may produce parallel changes. So we're seeing, just by developmental staging, an earlier change where vasculature doesn't develop. That's not affecting the pancreatic beta cell development. That's a separate process, but caused by the same original insults, the placental insufficiency. So I can't tell you the answer. I can only say that you're asking the right questions, and they are part of our research program. Maybe in a couple of years I'll have some more answers for you. None of this, of course, is clinical. Yes, people have tried to supplement pregnant women carrying IUGR fetuses with oxygen, and they didn't see any major results. Well, what does that mean? Breathing a high oxygen mixture is not good for anybody. Oxygen is essential to our lives, but it's toxic at only a slight increase. 23% oxygen is bad for you, just as 19% oxygen does things also. 21% is fairly tightly regulated. So giving oxygen to the mother hasn't been shown generally, although what were they following? They measured birth weight. Well, that's not a very good measure. So hopefully in the animal models we'll be able to show how we can independently regulate PO2 and oxygen content, which are very separate mechanisms in regulating blood flow to various organs and growth. And those are some of the things we could talk about for a long time in another meeting. Thanks very much for that. Good question. Yes?
2 practical questions.
Good.
First, what about the metabolic future of a healthy term, fully breastfed neonate who gains too much weight for 6 months, the first 6 months of life?
Yes, excellent question. I get asked this all the time. So if you're breastfeeding and your baby gets really fat, well, most breastfed babies actually put on fat weight. They go from, say, 10 to 15% body fat up to 20, some have even been up to 30% body fat. So as far as we know, that by itself happens slowly over time, and therefore is not part of a very rapid gain of excess calories, and those babies do not go on to have adiposity in their later life, different from the family structure. So if the mother is obese, the baby will tend to become obese no matter whether she over breastfeeds. I shouldn't call it over breastfeeding. That's just breastfeeding that leads to increased weight gain and due to adiposity in the baby. So those are independent factors, it appears, and that's the good news. So I don't think I would ever tell a mother, look, your baby's gaining too much weight, stop breastfeeding, slow down, because what are the alternatives? Vegetables?
No, but
Yeah.
which is the metabolic future?
Yeah, I think the metabolic future is good for those babies. Also, their intellectual and behavioral outlook is good. All right, both of them are good. So keep at it.
Okay. Second question, the opposite. We have IUGR, a premature baby, totally breastfed. He can't he hasn't a catch-up, and we cannot allow the discharge. He is 1,500 grams, and he can't gain a weight. He is fully breastfed and fortified. Can we add something? Carbohydrates or fat? Lipids?
I was listening very carefully, and I was going to try to go back to that 1,500-gram baby, that picture I showed you. So keep that picture in mind. That's the baby you're talking about. It's a term infant. Whether it was born preterm or not doesn't matter. That baby is severely, chronically growth restricted. And those babies do not have the same growth potential. Overfeeding those babies will only produce obesity, insulin resistance, and diabetes later in life. I think this gets back to the other question I had about that rat data, which suggests maybe these are the babies where we want to simply emphasize the quality of what we're feeding, so breast milk is good for that baby. And maybe we can look at those babies whose mothers can't provide breast milk and find out what's in breast milk that we could give them in a formula and do a better job of improving neuronal development, even if we can't get them to grow as well. I think the future lies in how do we find those babies before they get so severely chronically growth restricted, and how do we do a better job in the neonatal intensive care unit from preventing those babies from getting even worse, further growth restricted. But for a practical response to that particular baby, I would feed breast milk and allow the baby to take as much as it wants to take and encourage breastfeeding completely. For the preterm baby who's not growth restricted, there are very old data, a lot of them studies never published because it seemed obvious to the people who were doing these studies. I learned about this as a resident, so that was 100 and some years ago. From Dr. Lula Lubchenco. You maybe have heard of the Lubchenco growth curves from Denver, but she was my first attending. And she had a young mother's clinic, the first that I know of, where she had adolescent girls who had gotten pregnant and had a baby, and those that were moderately preterm, so what we would call late preterm infants, 32 to 36 weeks, when they became fully healthy and were taking full breastfeeding, they would take 200 to 240 cc's per kilo per day and achieve relatively normal growth rates. Now, that's a huge volume of feeding, and it didn't develop overnight. It developed over 2 to 3 weeks. But I think that's the alternative, just more of a good substance. Thank you.